Hello, everybody, and welcome to today's webinar by the Global Antibiotic Research and Development Partnership, GARD-P. My name is Astrid Pence-Moore. I'm a Scientific Affairs Manager at GARD-P, and I have the pleasure to host today's session on the topic of regulatory aspects of balancing benefits and risks in the clinical development of antibiotics. A little background on our activities. So REVIVE is one of the main outputs of the Scientific Affairs team of GARD-P. It is an education and outreach program which connects and supports the antimicrobial discovery, research and development community. Our webinars are part of our educational activities. As you can see on this slide, all webinars are recorded and can be viewed at any time after the webinar. Today's webinar will be recorded too, and it will be added to the website within approximately 1 week. Other types of content you can find on the website are our antimicrobial viewpoint articles, as well as the antimicrobial encyclopedia. I also want to inform you about the upcoming antimicrobial chemotherapy conference. This is a virtual free of charge conference which we are organizing together with the British Society for Antimicrobial Chemotherapy. It is taking place on 1st and 2nd of February of 2023, and this next edition of the conference is being developed in collaboration with REACT Africa and Africa CDC. Also, even though this is a virtual conference, you have the opportunity to present posters at the conference, and also 5 posters will be chosen for short oral presentations. So please make sure to submit your abstracts on the conference website as shown here, acc-conference.com, by 25th of November. Coming back to today's session, as always, you can ask questions in this webinar, and these questions will be addressed at the end in the Q&A session. But you can send your questions at any time by adding them in the questions window in your control panel. And if your question is for a specific speaker, please include their name when submitting the question. So now I have the pleasure to present today's panel. We have 2 speakers, Enrica Alteri, who is a former head of division at the European Medicines Agency, and John Alexander from the US FDA. And our moderator is Radu Botgros, who is a senior scientific officer and a specialist in infectious diseases at the European Medicines Agency. Radu has worked as an ID clinician for 10 years before joining the EMA in 2009 in the pediatric team. And from there, he moved to the anti-infectives and vaccine team, where he worked with the efficacy and safety related pre- and post-authorization aspects of centralized marketing authorization applications for the treatment and prevention of infectious diseases. Since March 2020, he's a member of the Health Threats and Vaccine Strategy team. Radu's main interests are with the development of antibacterial, antifungal and antiviral medicines, and with public health, with a special focus on the fight against AMR. Radu, you can switch on your webcam now. Yeah, thank you. And I'm handing over to you to introduce our first speaker, Enrica. Thank you.
Thank you very much, Astrid, and hello to everyone, wherever you may be. Good morning, good evening, good afternoon. Indeed, I have the pleasure to moderate this hopefully very interesting workshop organized by GARD-P. And our first speaker today, as you see on screen, is Enrica Alteri. She is a person that I know very well because she's been my boss at the EMA, and actually, you know, she has studied medicine in Italy, and then after she completed her medical studies in Rome, she went for a postdoc fellowship in the United States at the National Cancer Institute in Bethesda. She then, spent 25 years in the pharma industry, and actually she was involved in a number of major projects, and those projects yielded a number of important drugs that you will have heard about. I'm talking about Gleevec for myeloid leukemia, about Coartem for malaria, and Reatas for AIDS. And obviously, in 2012, as I said, she joined the EMA, where she was the head of the Human Medicine Division, and after that of the R&D Support Division. and a member of the EMA executive board. And obviously in these roles, she was part of really many high impact initiatives of the agency, including all what we do about AMR, about antibiotics in particular. She's currently a member of the scientific committee and faculty of the master course in drug R&D at the University of Geneva. And she also conducts master courses in R&D at the Luis University and Università Cattolica in Rome. She's recently been appointed core member of the UNITAID proposal review team, and in addition, she's a member of SCIOMS, of the working group Real World Data and Education. So without further ado, I'm very happy to welcome Enrica. So Enrica, please take it away.
Thank you very much, Radu. Thank you for the invitation, the introduction, and of course, thank you to GARD DP for inviting me in this webinar. My presentation will be rather short, and it will be really focusing on giving an overview of what balancing benefits and risk in the development of antibiotic means in regulatory terms. Of course, it's a complex problem and it needs really articulated solution. Today, we will not be able to touch on all the aspects of the work that needs to be done and is being done by regulators, and not only by regulators in this field. However, we hope to give you a feeling of the particular role that regulators should have, may have, and actually do have in fighting the big problem of antimicrobial resistance from their perspective. I was, as Radu said, a member of the European Medicine Agency, but this talk, it does not represent the view of my former employer. So I'm here in a personal capacity as an expert and as an interested party, if you will. The themes of my presentation will be rather, that's presented in this slide. So I will give a little bit of background just to give an introduction on the issue. I will touch very briefly on the regulatory environment and its complexities. I will present very briefly again recent guidance in Europe, in particular on development of antibiotic. And then I want to focus a little bit on what it means to have to develop an antibiotic and to find for this particular medicine the right benefit risk. This is not a medicine like any other, and we will see why. And then I will touch upon what is ahead of us in terms of R&D and the challenges that are involved. So antimicrobial resistance is a major threat. I don't need to tell you that. The audience is very familiar with the problem. By some estimates, the WHO recent estimates, in 2019, there were 1.27 million deaths due to antimicrobial resistance. That's, of course, a staggering amount, and is something that needs to be addressed. We all know also that there is precious little research in this field. Of course, there is effort, but as we know also, and this is maybe a separate conversation, the interest, particularly of pharmaceutical companies, to invest in R&D for antibiotic is relatively limited, because what we want ultimately is to develop medicines that we don't want to be used in large amounts and indiscriminately, if you will. So it is complicated, but recently, in the last few years, we have seen some revival of R&D, even at a basic level in this space. And this, of course, is encouraging. Why is it complicated, or is complex anyway, to do research in antimicrobial resistance? Because it's not just simply to identify a target and developing a potential medicine against that target. It really impinges on the complex environment whereby these products are not only used in individual patients, but they are used in animal for animal feed. So it is a vastly more expanded use, if you will, that is done and is being done across the planet. The individual country practices, both in terms of animal use and human use, vary enormously, and this has been one of the reasons why the antimicrobial resistance has been and is such a major threat, because there is no harmonized and unified approach on how to, if, when, and how to use which antibiotic in which situation. To complicate issues even more, just the identification of useful data to frame the issue, to understand what is the utilization of which antibiotic in which country, to which extent, and for what indication. This is not a simple issue to resolve. Again, in this space, there has been major effort done, particularly in the European Union, but elsewhere as well, to even point out and measure, quantify the extent of the use of antibiotic, which antibiotic, for what reason, and how are they actually used. So all this complicates very much the issue of research in antibiotic development, also from a regulatory perspective. And why? Because there are many players in this field. So we are not just simply talking about one regulator or another, or multiple regulators in various countries. We are also talking about individual countries prescribing issues. We are also talking about entities or in organizations that regulate and control the use of antimicrobials in raising cattle and animals for human food. This complexity, which I also mentioned before, requires the recognition by regulators such as the FDA or the EMA that there are various players in the field, and this is not just a binary situation in which a developer prepares a drug and a regulator approves it. I put this little note targeting a bug or a disease. Why? Because even in our more limited area of influence, which is the one of the regulated development of medicines, until recently, there were quite different approaches between, for example, the FDA and the EMA. So companies, even if they wanted to make, as they need to do, a global development, had to deal with a different or potentially not completely contradictory, but at least complicated set of guidance. Whereas for one regulator, the target unit was, for example, a condition, a disease, a specific infection of a specific organ, whereas for another regulator, the target was a microbe, a bug, an infectious agent. So this gives you the flavor that when a company wanted to do a global development for a new antibiotic, they had to kind of juggle the data, putting them in a way that would satisfy the needs of EMA on one side of the ocean and FDA on the other side of the ocean. This situation has improved dramatically in the last few years. Harmonization efforts not only are ongoing, but have already resulted in substantial changes in the guidance and also in the expectations the regulators have about what they see, in fact, in terms of data packages when they review a new product. The new EMA guideline has been out for a while and is coming into effect already now in December 2022. This is a very important development because, first of all, because there is a substantial alignment of the requirement with the US and Japan. I put here in the presentation, you will have it, the link to the guideline. In parallel, there is the pediatric addendum of the guideline, which has been revised and harmonized and updated. And John will discuss more in detail the pediatric requirement. I will not touch them. I just want to highlight one or two points of importance in this new guideline. There is particularly, there are lots of details in which situation about situations in which single pivotal trials may be accepted to support infection site-specific indications. The guidance on the presentation of the microbiological data, the clinical efficacy data, and the summary of product characteristics has also been revised. So I let you, go in more detail of the guidance. It does represent really a major conceptual effort to put together, bring together the knowledge acquired in the last few years about drug development, what works and what doesn't work, and also really has been listening to the input of stakeholders, company, but not only company, also WHO and other guidance in terms of what the requirements may be and should be. I wanted though to specifically, because it's also in the title of the webinar, what does it mean balancing benefit risk in terms of regulatory terms when we are discussing antibiotics? I remind you that for regulators, the benefit risk is a balance and is something which is patient specific. So when regulators review an application, they have in mind a patient with a condition for which the approval is sought or the marketing authorization is sought, and the data displaying or proving the efficacy in that particular condition should be outweighing. So the benefit for the patient should definitely outweigh the potential risk to that particular patient with that particular condition. That's the concept of benefit risk. Now, when we discuss antibiotics, this concept doesn't quite fit in the same manner. Why? Because the individual patient, yes, is the target of the therapeutic intervention. Also in the case of antibiotic, a person has an infection and a medicine is intended to treat it. However, the issue is definitely more complex. Why? Because the risks may not be only to that particular patient, but the risk as well as the benefits of the therapeutic intervention, the new antibiotic, may also be, or even are in certain respect, more relevant very much at a population level. What do I mean by that? So, for example, antibiotic resistance. Where do we put antibiotic resistance in the concept of benefit risk? Is it a problem? Is it a risk for that particular patient? Is it an issue of lack of efficacy? Is it a matter of lack of benefit for that particular patient? Or the use of antibiotic in this patient today, which may be effective and also safe, may represent a risk tomorrow for new patients, because it may engender resistance. So as you can see, this concept of benefit risk, which is very simple or quite basic in drug development in general, and that's the way regulators approach their review, for antimicrobial, it gets complicated, in fact, by the issue of resistance. So resistance is usually identified in the risk management plan among the risks. So it is supposed to be a problem and therefore a potential risk, but not necessarily for that patient, but for the population in general. More interesting is that even in more recent time, the potential for antimicrobial resistance is being identified and targeted as being a lack of efficacy that can touch an individual patient today. And of course, it will touch entire populations if this resistance develops in the future time. So ultimately, the benefit risk judgment and identification also not only affects a population down the road, but affects even an individual patient, because it does impact the choice that an individual doctor will do of the antibiotic for that particular medicine. So this is just to give you a flavor that in this big field of antimicrobial and anti-infectives, the issue of antimicrobial resistance is also dealt by the regulators, although they are not the only player, as I explained, in this very broad field, but is also addressed within the sphere of influence that the regulator have, to make sure that this is recognized and it is reported and is identified in the course of development, and of course in post-authorization. For example, in a recently approved antibiotic, the development of resistance should be followed in the periodic safety update report that are given to the authorities and to the EMA on a regular basis at regular intervals. So this one gives you the feeling of the overview that the topic of antimicrobial resistance and benefit risk of antibiotic is actually a little bit more complex than meets the eye. So what is the road ahead? Of course, we continue the efforts on all fronts. And again, from the regulator viewpoint, the efforts are well underway, and not only the harmonization has been ongoing now for years with the FDA, EMA, and the Japanese authority, the PMDA, but also more recently, other authorities, particularly in low and medium income countries, have been involved in having an active role into the understanding of what they can do as regulator to address in the most efficient way the issue of use of antibiotics and developing and use of new antibiotics. The WHO, of course, coordinates the One Health response. I want to flag here also that the 18th to 24th of November is antimicrobial resistance awareness week. So the WHO promotes a lot of activities in this space, and the promotion of R&D is one of the 4 strategic goals the WHO has also in this area. So it's very important that regulators, companies, and other instances, public or private partnership that develop antibiotic, we need to realize that we are part of a very complex infrastructure mechanism in which many players play a role. I want to just also mention that to fight infections, there are other approaches than antibiotics, and they need to be fostered in terms of research and development as well. It's not only vaccine, that's an obvious alternative, but there are others in terms of prevention and simply measures of hygiene and safe use of the available medicines that contribute to the overall goal of preserving the existing antibiotics and using new ones in the most appropriate way. So this introduction, and I hope I didn't speak too much, is really means to get your flavor of the fact that yes, developers in companies, in other public-private partnership organization, in academia, they work on new medicines, and any R&D effort is very welcome and necessary. But it's also important to maintain the big picture of what is the whole field all about. So with that, I hope I gave you whet your appetite for either more detail or more general discussion, and I pass it to Radu. Thank you very much for your attention.
Thank you very much, Enrica, for this very nice presentation, and some of the ideas that you mentioned here are very important, food for thought for all of us, and obviously may well be discussed after the end of this presentations. And just allow me now to introduce to you our next speaker. It's Dr. John Alexander from the FDA. He's currently Deputy Director of the Division of Pediatric and Maternal Health in the CDER, so the Center for Drug Evaluation and Research at the FDA. John's a pediatrician who joined the FDA in 1995, at the time as part of a joint fellowship in pediatric ID with the FDA and the Children's National Medical Center. And after he completed his fellowship, he became a full-time medical officer and subsequently team leader in the division of anti-infective products. He's worked so far in the review of antibiotic drugs and pediatric drug development for more than 20 years. So welcome, John, and over to you.
Thank you, Radu. My talk, I want to start out by thanking Radu for the introduction, thanking the staff at GARD-P for inviting me to speak today. My presentation is going to focus more on safety considerations in pediatric antibacterial drug development. As was mentioned, I'm the Deputy Division Director for the Division of Pediatrics and Maternal Health at FDA. So a lot of my current focus for the past 5 years or so has been more on pediatrics in general, but I do have a fairly long background in antibacterial drug development. So I did want to show my disclaimer. This presentation reflects my views and should not be construed to represent FDA views or policies. I don't spout FDA policy very easily, so I want to make sure that you understand this presentation is based on my experience, and I have no financial conflicts related to this presentation. So as an outline of what I'm going to talk about, I wanted to start the issue of looking at safety considerations for pediatric antibacterial drug development with a focus on the framework in the United States that I work under for pediatric drug development in general. I'm going to touch a little bit on adult antibacterial drug development, and then speak about pediatric extrapolation for antibacterial drugs and what the considerations are there before getting into more detail in the discussion of pediatric antibacterial drug development and studies needed. So starting out with the pediatric drug development framework in the United States, and I'm going to give you a brief overview. I hope this isn't too simplistic for those of you on the phone today, but I think that there are some general principles that we need to keep in mind that apply. And these principles are outlined in the E11 guidance for clinical investigations of medicinal products in the pediatric population. So the idea that pediatric patients should have access to products that have been appropriately evaluated, and product development programs should include pediatric studies when pediatric use is anticipated. I think that that's important to think of even for these drugs that are being developed for treatment of resistant infections, because even though they may be more rare than the general types of infections due to susceptible organisms, it is still expected that such antimicrobial resistant infections will occur in the pediatric population. And the whole reason that you're developing the antibacterial for treatment of drug resistance in the adults is applicable to the pediatric population as well. So in the US, there are two main pediatric legislations that affect the development of drugs for pediatric populations. The Pediatric Research Equity Act, which requires companies under certain circumstances, which I'll outline on the next slide, to assess the safety and effectiveness of new drugs or biologics in pediatric patients. And then there's also the Best Pharmaceuticals for Children Act, which can provide a financial incentive to companies to conduct pediatric studies to support the use of the drug in the pediatric population. I'm not going to discuss much more about this, but I did want to mention that part of our providing a financial incentive to companies is to look at all of the potential uses in the pediatric population for a drug. So when it comes to antibacterials, I do think that there's a tendency not to be able to issue a written request in under BPCA and offer the financial incentive, in part because of the fact that there's such a focus on a single indication that a drug is being developed for, and not the consideration of sort of the broader potential uses. So we're oftentimes unable to issue a written request because the company is focused on just one potential indication that's being studied for a new drug rather than other potential indications. So then in terms of the PREA requirements, PREA gave FDA the authority to require pediatric assessments under certain circumstances. And the idea is that data gathered from pediatric studies using appropriate formulations to assess safety and efficacy and to support dosing administration of a drug or biologic product in a relevant pediatric subpopulations for the same indication for which it's being sought in adults, unless that requirement is waived or deferred. So in most instances where we're talking about general infections like community-acquired bacterial pneumonia or skin and skin structure infections, complicated intra-abdominal infections, those conditions occur in the pediatric population too, and so it's important to consider the pediatric population that may benefit from the availability of this drug for treatment. Under the PREA requirements, FDA may also require companies to develop age-appropriate formulations to conduct required studies, but does not require those companies to necessarily market that formulation. I think that's an important consideration, particularly for oral antibacterials, but a lot of current antibacterial drug development is focused on use of intravenous products, and so this becomes a little bit less of a concern. Those intravenous products, injectable products, can usually be diluted appropriately to be administered to the pediatric population. PRIA provides specific criteria for when study requirements could be waived. I think that's also less of a consideration in the antibacterial space, where infections that occur in adults also seen in the pediatric population. PRIA does not apply to products that are granted orphan designation, except for a particular exception for molecular targets in oncology drugs. I won't go into this further, and it's very unusual that a product that's intended for treatment of infection would necessarily qualify for orphan designation. One of the important parts of PRIA that's very helpful in pediatric drug development is the initial pediatric study plan, which was initiated around 2012 and allows for earlier communication on pediatric product development, typically around the end of phase 2 for the adult product development. And I'll get into that a little bit more here, where I'm starting to talk about the antibacterial drug development in adults. So for adult antibacterial drug development, one of the things that I wanted to point out is that generally for antibacterials that are being considered for studies in the pediatric population, you will have some adult data. It's very pediatric specific antibacterial drug development is fairly rare. There are a few indications where, such as otitis media, where efficacy may need to be established in the pediatric population because the disease differs considerably from what occurs in the adults, but for the most part, you are relying on data that are gathered in adults with an infection to establish the efficacy of the drug, and then the question is what's needed in the pediatric population. So what I'm going to go through on the next slide is some of those aspects of the adult development program that support pediatric drug development, because that's the focus here. What is it that you're getting out of the adult antibacterial development that is an important consideration to support the further pediatric drug development? So one of the things that that we get out of the antibacterial drug development is knowledge of the drug product itself, including the active ingredient and the excipients. It is important to think about whether the drug product that's under study is something that's related to and in a similar class to the drugs that are already available, or if we're really talking about a new molecular entity that's really targeting a new target within the bacteria. The excipients are important because in the development of some of these antibacterials, particularly ones with issues related to solubility, the excipients can be important in the pediatric drug development, and there may be safety issues that need to be considered with regards to the use of some of these excipients, either because of a lack of data or the potential that an excipient may cause harm in the pediatric population that it wouldn't be expected to cause harm in the adults. One of the other parts that we get out of the adult antibacterial drug development is the knowledge from non-clinical toxicology studies that have been conducted those non-clinical toxicology studies are typically conducted in different animal species and provide some information on the potential safety concerns that may arise with the use of a drug. The animal models of infection are also important. Oftentimes, what you have as the basis for dosing of drugs in a population is information from animal models of infection that tell you what types of exposures may be needed in order to be effective in the adult studies. Although for the most part, what we typically expect to see is information from adult phase 2 or phase 3 studies that provide information on what the appropriate dose of an antibiotic is to be effective in a particular indication. We have adult phase 1 and phase 2 studies that provide us some initial information on safety and understanding of the ADME of the drug, and that is important because the understanding of the absorption and the metabolism of the drug may be very important in sort of deciding what types of trials might need to be conducted in the pediatric population. And finally, the adult phase 3 trials themselves. Most often, this is the main basis for the support of the efficacy of a particular dose that's intended to be studied in the pediatric populations. So understanding which indications are being studied for a particular antibacterial in the adult population is important. It also can affect how you go about conducting the pediatric trials and when you enroll pediatric patients with certain types of infection, and I'll get into that a little bit later in the presentation. So this is the point where I want to talk a little bit about pediatric extrapolation for antibacterial drugs. So pediatric extrapolation in general is based on two main factors: the similarity of the disease in the adult and pediatric population and the expected response to treatment. One of the benefits in terms of use of extrapolation in the pediatric population is that we have an understanding of the antibacterial effect of most drugs, and this antibacterial effect is based mainly on a direct effect on the pathogen that causes the bacterial infection and is independent of whether the adult is the host or the pediatric patient is the host of the offending pathogen. So the expected effect of the drug is still going to be the same, and in fact, any adult or pediatric host interactions are usually related to concerns about adverse effects or side effects of the drug in the population. So for the most part, we're looking for antibacterial drugs that have an effect on the pathogen, but don't interact for the. We're looking for drugs that won't have much of an effect on the host themselves. So when it comes to pediatric extrapolation for antibacterials, for the most part, we're comfortable with the idea that you can extrapolate the efficacy of the drug from the adults, and what you're mainly looking then for in pediatric patients is in pediatric studies is generally information about safety and the pharmacokinetics of the drug. I will mention also here one of the considerations for extrapolation that's fairly new, this ICH E11 a draft guidance talks a little bit about extrapolation of safety. I want to touch on this concept a little bit. The main idea, or the way that I think about this, is mainly the idea that you do get a certain amount of information from adults about what adverse effects are known to be seen, especially once you've gotten information from phase 3 trials in adults and you have an understanding of the adverse effects that are associated with the drug. Those are the adverse effects then that are important to be studied and evaluated in the pediatric studies to make sure that there isn't a great difference in the frequency of those types of adverse effects in the pediatric population in comparison to adults. So that's the way that I think of this extrapolation concept for safety purposes. You are basically taking the information that you're getting from adults from the non-clinical studies that have been conducted to support antibacterial drug development in the adult population, and using that to try and make sure that you have a good understanding of the types of safety concerns that you might need to think about in the pediatric population. So let's move on to some points with regards to the pediatric antibacterial drug development. So the first thing to consider when trying to design pediatric trials for a particular drug is the pediatric population that should be included in those studies. And typically this is based on the epidemiology of the infection that's being studied. I mentioned this before, but I noted here that the difficulty with regards to design of some pediatric trials, it can become more complicated when adult studies for some indications are planned as supplements to an original application. So, for example, a company is coming in and they want to conduct as part of their original application studies of their drug for complicated urinary tract infections, and that's going to be the first indication that they're going to seek in the pediatric population. But then they plan to follow those shortly afterwards with other studies in, say, complicated intra-abdominal infections or in hospital-acquired bacterial pneumonia, ventilator-associated bacterial pneumonia, and plan on seeking those indications in supplements that follow. The issue then gets to be about the pediatric studies, because the way they may need to be designed is designed around initially evaluating only those patients with complicated urinary tract infections, because that's the place where you have information already about the safety and effectiveness of the drug, and you have the information about the dose that's shown to be effective in adults that you're then extrapolating to the pediatric population. So this can become complicated at times and does affect the pediatric trial when you're then waiting for additional studies that are being conducted in other indications, especially if those indications involve different doses or slightly higher doses of your antibacterial drug that you're evaluating for treatment of those different infections. I want to move on then to talking about how you go about studying certain populations. And one of the things that we are focusing on, because we are trying to focus on earlier availability of information for the pediatric population when possible, one of the things that we want to focus on for antibacterial drug development is the consideration for inclusion of adolescents in adult trials in certain circumstances. One of the reasons that we do this is because typically adolescents will receive the same fixed dose as adults, and for the most part, older adolescents are within the same weight range as we see for adults in adult studies. And so it makes sense that if a fixed dose is expected to be used for adolescents that are, let's say, above a 40 kilogram threshold, that those adolescents then can potentially be enrolled in the trials for adults that are being designed, and that would then allow for us to have earlier information on the safety of the drug in the adolescent population in order to include them. Now, the first sub-bullet there, prospect of direct benefit and risk-benefit considerations, as well as practical aspects to the inclusion of adolescents, may preclude this. So some of the considerations that go on there are, if you're really dealing with a new drug and you have particular concerns about particular safety concerns for a drug, it has effects on liver toxicity, it has certain effects that may raise questions about the risk-benefit, then the inclusion of those adolescents may be more difficult in those studies. But I also think that for the most part, if you've decided that you have adequate information to study a particular dose in the adult population, that really is the that information should be adequate to also then support, in most instances, that the adolescents would have the same prospect of direct benefit. I do mention in this bullet the idea that there are some practical aspects to the inclusion of adolescents in adult trials. Some things like, trials that are being conducted at certain sites where they really only see adults and don't have significant adolescent populations. So you really have to sort of think about some of those aspects if you're going to be considering the inclusion of adolescents in an adult trial. There may need to be inclusion of children's hospitals as some of the sites to conduct the studies in order to get sufficient adolescent populations, or sites that have both adult and pediatric patients that are seen at those sites in order to be able to enroll them. A couple of the things that I think are important to think about if you're going to enroll adolescents in an adult clinical trial is that we would still typically look for some confirmation about similar exposure. So doing some sparse PK sampling in the adolescent population that is included in the studies may be helpful. And one of the considerations, if you're going to decide you are going to include adolescents in a population, is thinking about what safety evaluation is planned in this subgroup. So it's a consideration not only about how many adolescent patients are you expecting that you will be able to enroll within the trial, but also is there a specific plan for evaluating the safety within that subgroup so that you can know that what you're seeing is similar to the adult population that's enrolled in the trial. And then one of the other considerations here is that if you move forward with enrollment of adolescents, you may need to be ready to conduct additional studies in this age group if there happens to be low adolescent enrollment in your clinical trial. And I have seen several examples of this even in the antibacterial drug development space, where a sponsor had planned on being able to enroll a targeted population of let's say 40 or 50 adolescents in the trial, and they got 2. So the idea that you may need to be ready to conduct additional trials in the adolescent population as part of the other pediatric studies is important. So what pediatric studies are usually conducted for the younger population, those that are sort of below the weight threshold that is typically used for enrollment in adult studies. So once you get into the population where you may need to adjust the dose from the fixed dose that you plan for adults to starting to give weight-based dosing, one of the things that's oftentimes done is single-dose PK studies. I would emphasize that these should be considered only when needed. There are many drugs where, and the considerations here, I'm sorry, are that you really need to think about the absorption, distribution, metabolism, and excretion of the drug and what you have available to you in terms of PK modeling. A lot of the antibacterial drugs that we see don't really have any substantial metabolism and are, for the most part, renally excreted. And for most of those drugs, we're able to be fairly successful at using PK modeling to predict what would be the appropriate dose of an antibacterial drug to use in the pediatric population that matches the adult exposure that you're targeting. I think I would recommend that single-dose PK studies be more reserved for those situations where you do understand that there's some metabolism of the drug and that the exposure may change in certain age groups, and the target for the single-dose PK study really ought to be in sort of understanding the potential changes in the pharmacokinetics in those populations. That's usually the younger population, less than, say, 2 or 3 years of age. My talk was supposed to be focused on safety data, so I don't want to spend too much time on the single-dose PK studies, in part because we don't get a lot of safety data from those trials, especially when you consider that these single-dose PK studies are typically conducted in pediatric patients with a particular infection that are receiving typically multiple doses of a standard other antibacterial drug and are receiving this single dose of this investigational treatment as part of that treatment course for that infection. So we don't really get a lot out of the safety data from these types of trials. So you have to conduct then multiple dose pediatric studies, and the pediatric study should be designed around the evaluation of the safety of the product given at the dose and duration that's intended for clinical use. And this is important. For the most part, you're talking about, for antibacterial drugs, something that's a fairly short course treatment, 1 week, 2 weeks, something around there. But there are antibacterial drugs that are required to be used for longer periods of time. Examples are the drugs for tuberculosis. And in those instances, you may need to consider whether there are studies that need to be conducted, such as juvenile animal studies, to support the safety and the evaluate whether there are any effects of the drug on growth and development that may be detrimental to its use in the pediatric population. For the pediatric studies, where we're typically looking at safety of a fairly short-term exposure of a couple of weeks, you are focused on exposure matching to the effective dose in adults. And so this is oftentimes why we're waiting to see the results of phase 3 trials in the adult population to understand both the dose that's considered to be established to be effective in the adult population, as well as the as well as an understanding of the risk benefit overall. Staggered enrollment in these pediatric trials by age group is often done. I would say that it's not always needed, especially, again, going back to what I discussed before with regards to drugs that are known to be renally excreted, that you can effectively model what doses and exposures are expected to match those in adults. There really isn't a lot that you get from sort of looking at safety in, let's say, 6 to 12-year-olds that then inform you for sort of going down to the next staggered cohort of 2 to 6 years of age. So I do encourage sponsors to really think about what they're doing in terms of staggering enrollment for studies, because oftentimes it's not needed. So I'm getting towards the end of my talk. One of the things that I think is important to consider is the inclusion of the neonatal population. One of the difficulties here more often is the determination of the dosing regimen for term and preterm neonates. Again, as I discussed before, one of the safety aspects here is careful attention to understanding the excipients in the product. Benzyl alcohol is an example of something that can be very, very toxic to the neonatal population, but there are other excipients as well. Things like alcohol used sometimes as an excipient for particular drug products that that have difficulty with solubility. And those types of excipients can be harmful. You need to think about whether you can actually formulate an antibacterial drug in such a way to remove excipients that may be toxic or haven't been adequately evaluated in the pediatric population. One of the other things that I wanted to point out here too is, because of the difficulty in determination of the dosing regimen, these studies oftentimes in neonates are conducted separately, and I think that that can be important to making sure that you're evaluating both the safety and the pharmacokinetics of the drug appropriately in that population. I will note that we have a guidance, and I'll talk about those on the next page, that try and address the PK in this population. One of the other issues that I wanted to talk about with regards to the pediatric studies is just the safety concerns that are intended to be evaluated in the pediatric population. You need to really think about what safety monitoring that you're doing in the pediatric population, whether it's appropriate. I see a lot of things sort of copied from adults and expected to be conducted in the same manner for safety monitoring for things that just aren't possible in the pediatric population. I noted specifically laboratory cutoffs that should be age appropriate. I've seen examples of studies done where a sponsor became concerned because what they were seeing was, or what they were reporting was, hyperphosphatemia that was occurring in a pediatric population. And the whole issue was that they were using the adult laboratory cutoff for phosphorus levels for the pediatric population that was enrolled in the trial, when they were just seeing what would be normal variations in the phosphorus levels in the pediatric population. So you really need to think about what you're looking at for safety in the pediatric population. And I referenced several guidances here that I used within the talk. I wanted to point out in particular the first one, the development of anti-infective drug products for the pediatric population. This is a fairly new guidance that I think may answer some questions that you may have about pediatric population studies. I do note there's this pediatric extrapolation ICH E11A document that's now available as draft guidance on the FDA's website. I'm sorry I didn't include the link, but if you use the other links and get to the search page and just look for E11A, you should be able to pull up that guidance. Some of the other guidances that I just wanted to point out are the general clinical pharmacology considerations for neonatal studies. This is a relatively new guidance that may be helpful. And then the last one also, the guidance related to ethical considerations is new and I think is worth reading. Thank you.
Thank you very much, John, for this very good and exhaustive presentation about how pediatric drug development is considered by the FDA. And I would like now to tell the audience that we will start the Q&A session. Just as a quick reminder for you, you can submit your questions as shown on this slide, and we will do our best to respond to as many questions as possible during the allocated time. We have already coming in a number of questions, and I will start with one for Enrica. And the question is, could you reiterate what you talked about on recent harmonization developments with regard to the EMA FDA in terms of addressing the complexities of antibiotic drug development? Enrica, over to you.
Thank you for the question. I think is a question that actually, Radu, you are in better position to answer than I am, but conceptually, what I mean is the fact that the data package that is required by a regulator like the EMA and the FDA are now coming to be much more convergent. And I think John illustrated many examples of this convergence that is happening in practice. And I think the new EMA guideline that is coming into effect in December also is an example of that. And perhaps, Radu, you are better positioned to expand with concrete example than I am. I'm a little bit out of practice.
Yeah. Thanks, Enrica, for this. Actually, yeah, indeed, what I can say in addition to what Enrica has just stated is the fact that indeed we have been working with our colleagues in the United States and in Japan. We have had a number of tripartite discussions aiming at trying to harmonize as much as possible, the main pillars of drug development for antibiotics. And here I'm talking about, primary endpoint, a non-inferiority margin for non-inferiority studies, these kind of things. And I think the current version of the guidance, as Enrica says, the one that has been adopted by our scientific committee in May of this year and will come into force on the 1st of December, is actually already implementing the agreement that we have reached in these tripartite discussions. And just to give you an example, we had for complicated urinary tract infections, which is a fairly common indication, different primary endpoints, microbiological endpoints for these across across the pond, actually. And we have agreed with colleagues in the United States that indeed this was not something that was entirely justified. So we have agreed to converge, and this is just one of the long-hanging fruits. We will be continuing, and we are continuing these discussions with the Division of Anti-Infectives at the CEDAR, we have to work together to harmonize as much as possible. As Enrica said in her presentation, AMR is a global threat. Obviously, we need to involve not only actually the stringent regulatory agencies, but also as many as possible agencies from around the world, because AMR does not respect borders, and therefore, we can do as much as we can do, but we need to help others, go the right path, and this is what we are currently exploring. We have the example of the ICMR, which is the International Coalition of Medicine Regulatory Agencies. This could be one example where this could be further explored, to bring in regulatory agencies from other parts of the world as well. So I think that said, obviously there are some differences that still persist. We will have to see how best we can harmonize, in view of our mandate and of the legislation that's ongoing. I'll stop here because I see there are a number of other very interesting questions, and the next one I think is for Dr. Alexander. So I think it's pertaining, in a way, it's a continuation of the question that was asked for Enrica, and it says, can you comment on the above question, so i.e. the harmonization, in the panel discussion with regard to historical FDA perspective? I personally don't exactly know what is meant by that, but I'll over to you, John. I suppose it's about the change in perspective from the FDA side over the past decade, I suppose. Thank you.
Yeah, so I guess I will say that when it comes to antimicrobial resistance, since I've been in the pediatric division for many years now, I've sort of lost touch with what's going on with regards to looking at the questions of antibacterial resistance and antibacterial drug development that's intended to sort of address those issues with resistance. But I guess I will say that that is still, I think, something that was a focus even in the past, trying to understand what's needed to look at these drugs. The comments that Enrica had made previously about the considerations for how we go about developing these drugs that are intended to address these resistant infections, and then keep their use low because we don't want them to sort of be used indiscriminately or widely for treatment of general infections, and then you lose the benefit of the activity of the drug on resistance is something that has concerned the FDA for a long time as well. I think that when I was in the division a while back, one of the concerns that we had really was around using non-inferiority studies and understanding sort of how those non-inferiority studies are going to contribute to our overall understanding of the effectiveness of a drug. And that's why we had a lot of difficulty in trying to design what the appropriate antibacterial drug study in adults would be. And I think that the efforts that you mentioned, both about harmonization now, are trying to recognize that, that we have to think about exactly what kinds of studies that we're going to be doing, and that these studies are going to need to be multinational, international, and so trying to address harmonization in the way that you're doing with the Division of Anti-Infectives is important.
Thank you very much. And indeed, I think as well, there have been these changes in the optic with regard to, I think there were in the past some considerations about how to define the lower bound of the confidence interval, and, how you could justify non-inferiority margins so that you ensure that, the candidate drug is effectively better than placebo. This is something that is obviously very important whenever you are looking at non-inferiority trials. There is another question that says, what is the threshold of risk score for AMR, and what is the measurement tool? And if there are any lead references or studies that one could refer to, maybe I can start on this one. So I think I don't think we have a score to establish the risk. You know that once you start using antibiotics, you develop resistance, and obviously what we want to see in the dossier for for a new antibiotic, for example, is a number of things and tests that aim at looking at the potential of developing resistance. So if, for instance, we have a test drug of a new class, we expect that the in vitro susceptibility studies assess the potential for cross-resistance to occur between the test agents and licensed agents of other classes, whereas for test drugs of existing classes, we want in vitro susceptibility study to document the extent of cross-resistance within that class. We also look obviously at the frequency of selection of resistance, and that can be initially estimated by exposing strains of species relevant to the indication, so to drug concentrations below, at, or above the MIC. And I think it's what we also recommend, and all these are already in our guidance, is that we recommend that the risk of selecting resistance is also evaluated in an in vitro PD model, which uses drug concentration profiles that mimic those achieved or predicted in infected patients. And this is obviously pre-approval, but after approval, the requirement that we have for our marketing authorization holders in Europe is, looking at emerging resistance, obviously, is that they should promptly notify us whenever they sense a change in the patterns of resistance of the, or new mechanism of resistance of the antibacterial agent. So we sometimes we assess these changes, and sometimes we change the summary of product characteristic. But obviously, since some of those are quite important and urgent, sometimes direct communication with the regulators is needed, depending on that perceived urgency. So I hope I have addressed, and in terms of references, obviously, I would like to recommend that you read our guidance, because obviously we have all these requirements very well presented there. I don't know if any of you wants to interject here on top of what I said. Enrica, you are muted, yes. I think.
Yeah, I just wanted to underline that even what you just mentioned, it goes quite beyond the traditional role of the regulator. So it's a very multidisciplinary approach that both the regulator need to take and the company. So you have a number of tests and even approaches to look at a specific problem of an individual medicine that really now are expanding quite substantially the toolbox that we need to have for assessing the data, and that companies or developers need to do to present to satisfy the needs not only of the regulator to approve a specific product, but to really utilize the product in the most appropriate way on a more global scale. So I think what I want to highlight from your considerations and what John said before is that really we have entered a phase in which the regulator needs to be a little bit more multitasking and even collaborate with agencies and organizations that were a bit outside of our radar screen until only recently.
Yeah, indeed. And, just to say, as you were speaking, I also want to add that obviously what's also important when looking at AMR is the dose, the appropriate dose to be given. And obviously here we have the PK/PD tools that underpin, the appropriate selection of the dose. In particular, if you have a situation where, for instance, in particular for pathogen-specific indications where we will not have enough data from clinical trials with regard to those bugs that are resistant, there the PK/PD tool and the robust package that underpins the right dose is essential, because obviously if you give the wrong dose, the development of resistance is around the corner. So with that, I think I will get to another question. This one is for John. Could you please comment on PRIA's mandate to conduct studies in children, and if it also mandates companies to register this indication for label updates?
So the mandate is only applied to drugs based on the adult indication that's pursued. So in the extreme example where you say a product is being studied for adults and you start planning on pediatric trials and you get through the phase 3 and the product is not registered because there was some safety issue or something that failed within the trial and so it wasn't demonstrated to be effective. In those instances, there's no further requirement for them to study anything in the pediatric population because it actually has to be registered for the use in adults in order for the PRIA requirements to apply. That said, we're usually with antibacterials in the position of really doing confirmatory studies of the effectiveness of the drug in the adult population. You're just confirming that that drug dose was actually effective in patients with infections that were susceptible to that particular antibacterial drug. And so once the application comes in, the indications that were being pursued are those that face a PRIA requirement. And that's where the part of my discussion with regards to pediatric studies comes into play, because if you come in with an original application and you're only seeking complicated intra-abdominal infections in that original application, and then you're planning on coming in with supplements, but those studies aren't done yet, you really only have a requirement at that time for the original application to do studies of the complicated intra-abdominal infection. And if we don't have the information yet about the appropriate dosing for HAP or CUTI or whatever other indications that you're coming in with subsequently, then it becomes difficult to do a pediatric study and you may need to focus initially on the CIAI patients and wait until you have the additional information from those other adult studies in order to then support expanding the pediatric study to include those other patients with other infections.
Thanks for that, John. And I think the next. Sorry, please.
Can I make a consideration? I think also it's important that the regulator realize that all our efforts are absolutely fine. Essential is the cornerstone of everything, but we need to engage a lot more with healthcare professionals, pediatricians and GPs, because at the end of the day, they use the drug, misuse the drugs. I mean, we have ample evidence, particularly in some European countries. The one where I'm at the moment sitting is a beautiful example of improper, not correct use. So when we discuss about the dose, we are evidence-based, and then the medicine goes in the hands of practitioners that not necessarily really pay attention to what we say and to general problems more having to do with resistance more in general. So I think part of our role has become the one to engage with healthcare professionals and be sure that what we come up with and what the data says is actually translated in clinical practice correctly. Otherwise, what we do, it remains in the pieces of paper that we put together in the packages. And I just wanted to do this consideration because, we've seen it too many times. The data says that, what we do is perfect, and then real life defeats our own work. So just
and
to make,
to
sobering argument.
Enrica, I think, on the upside of what you just said, we at the EMA for sure, and I'm sure also FDA colleagues, but we at the EMA, we are interacting now with patients, with healthcare professionals, organizations throughout Europe, quite in an active mode. I can tell you last year we had a number of workshops with them in which we tried to inform them about what we are doing, and also want to hear from them how we can further support their practice so that indeed, the antibiotics are used as they should be used within the terms of the authorization and not in a different way. John, if I can ask you, you mentioned the PRIA requires companies to develop age-appropriate formulation to conduct required studies, but does not require companies to market the formulation. So one of the participants asked, who would then be marketing this pediatric formulation when approved?
So that gets to be sort of a difficult issue, and we sometimes have sponsors. I'm thinking of several examples, although it's out of the antibacterial space, so I don't know that it's as relevant here. But examples of formulations that were developed specifically to sort of study younger patients with a condition that occurs commonly in adults but is very rare in the pediatric population. So they go through the process of actually developing, they submit the studies to their NDA, but then they decide after a while that they're not going to market the pediatric formulation because it's just not big enough of a market for it to be worthwhile for them to do so. We have difficulty then in those instances, especially in certain circumstances where those studies might have been conducted in response to a written request and they actually got some sort of additional exclusivity for conducting those pediatric trials, that they then decide that they're not going to market those drugs. But again, this is an issue where we have limited authority because the authority actually allows us, or what we've interpreted the legal authority is that we have the ability to sort of require these drugs be developed so that we can study them in the pediatric population. We don't necessarily have the authority to say you must market it.
Very much for this, and I think we are coming close to the end of this webinar. I'll just take one last question, which says, given that animal husbandry is a source of antibiotic resistance, how do regulators address the simultaneous application of a new antibacterial for both human and animal use? And if you agree, I will start answering this one. Actually, we all agree that AMR is a One Health topic and therefore needs a One Health approach. In Europe, you know that we have recently worked together with our vet colleagues on developing a list of critically important antibiotics reserved for human use. And this is probably a more complex discussion, but just to give you a bit of a flavor of the work, I mean, we have started from the important lists of critically important antibiotics that are out there already, the WHO one, the CDC one, and actually we have gone through each and every class of antibiotics and have assessed it for criticality of use in humans, for the potential of developing and transmitting resistance, antibiotic resistance, and looking also at the use in animals and the importance of that use. And here I'm talking about obviously therapeutic use and prophylactic, metaphylactic use, and not about feeding, as feed help for growing animals, because this is banned in Europe since quite a while now. So we have therefore this list, and just to answer this very question, I mean, new antibiotics definitely are on that list, so are deemed as critically important. We don't have all that many, unfortunately, as we all know, and importantly, I think they also, I mean, one needs to say they are also quite pricey, so I suppose that their use on large scale in animals would anyway be a little bit prohibitive. But just to reassure you that we as regulator, we are looking into this, and we have this tool that now will be revised. We don't yet know how often, but it will be revised most likely, so that any new class that emerges, and we do hope that they will emerge, will make it on the list. I will stop here, and obviously now I will give the floor back to Astrid. I want also to thank her very much, also on my behalf, for allowing me to moderate this very interesting discussion. So Astrid, over to you.
All right, thank you very much. So thank you, Radu, for moderating the session so well and discussing also so actively in our Q&A. And thanks, John and Enrica, for your presentations and participation in the Q&A. I will now take the opportunity to announce 2 upcoming webinars. So on the 22nd of November, we have a GARD-P webinar on the topic of putting children first towards a better outcome for children and newborns with drug-resistant infections. So you can already register for this on the GARD-P website. It's gardp.org. And soon also we will add the information on the Revive website. And following that, on the 13th of December, we will have another Revive webinar on the topic of influence of biological sex in infectious disease. And this webinar will soon be added to the Revive website for registration. And also all participants of this webinar will anyways get an automatic follow-up email in a few weeks with the registration details. Once more for those, but in brief this time, for those who joined later, we also have a virtual conference coming up in February, for which I would urge you to register for. It's for free. And also you can submit poster abstracts for this conference, despite it being virtual. And also some of the posters will be chosen for oral presentations. So I think that's a really good opportunity for not only, but also for early career researchers. So I would really recommend you looking into this. And with this, I'm at the end. So thank you, everybody. Thank you for joining and participating. And I hope you all join for future webinars and spread the news and the information with your colleagues and network. Thank you very much, and goodbye, everybody.
Thank you very much. Bye-bye, everyone. Thank you very much. Bye.
Thank you.